Hello, everyone. Good afternoon, good evening, and good morning. So it's my pleasure to introduce you today, our speaker, Dr. Daniel Irimia. Dr. Daniel Irimia is a friend and a colleague at Massachusetts General Hospital and Harvard Medical School, Center for Engineering in Medicine and Surgery. So Dr. Irimia is an associate professor at the Harvard Medical School, and he defines himself as a bioengineer, a medical doctor by training, and a researcher in the areas of microfluidics, inflammation, and sepsis. Dr. Irimia started out his training in Romania and then moved to the United States, and then became what he is. So he's the person to talk about neutrophil behavior in different disease settings, because he's been using his microfluidic technology to study the behavior of these cells and in different immune settings. So he was recently awarded Pioneers of Miniaturization Prize from the Chemical and Biological Microsystems Society for his work on these microfluidic tools to analyze neutrophils and other leukocytes in health and disease settings. So today we're going to be doing a little bit differently the questions. So Dr. Irimia has divided his talk into parts. So we'll be stopping after about 15 minutes into his talk where he will address the questions. So please bring in your questions in the Q&A as always, and we'll address them during the talk. So again, welcome Daniel, and we're very excited to hear your talk.
Thank you very much, Başak, for the invitation and for the introduction. And
all right.
And so I know this is one of the last presentations of the semester, so I thought I would make it a little bit more entertaining by showing you a lot of movies. And I also structure the presentation such that we have 2 breaks before the end where you can ask questions. And I would encourage you to think about questions as we move along the way and post them, and we'll stop after the first 15 minutes and then after half an hour. And I'll take 1 or 2 questions and answer, just to make sure that everyone is still awake and engaged with what I'm presenting. So if I don't get any questions, I'll stop. Today I'm going to talk about neutrophils, about chain reactions, about neutrophil swarming. And as I told you, I thought about this presentation as a movie. So let me introduce you the actors first. Neutrophils are the white blood cells, will be the main character in this movie. And then I'm going to talk about microbes, which are, in this case, bad guys. And they have funny names like Candida albicans, Aspergillu, Staphylococcus. Some of you might have heard about this. The conflict between white blood cells and microbes in the context of the body and human health is called infections. But in our case, as engineers, we are going to look at these interactions in a more controlled setting. We build microfluidic arenas, and I'm going to introduce you to this new way of looking at infections and learning about infections using engineering tools. So neutrophils are the cells that destroy microbes and clear infections. And you take any book that has a chapter about immunology, you'll see this picture about neutrophils, how they look in blood with this special nucleus that is divided into lobes. So neutrophils are most of the time in the blood. We have about 25 billion neutrophils in the blood. And whenever there is an infection in a tissue, neutrophils move from the blood through a process of transmigration through the endothelial cells into the tissue where they encounter microbes. They interact with other immune cells and they create a complex immune response. When I started my career looking at neutrophils, I thought that this imbalance between how much emphasis is put on neutrophils in the clinic, where physicians want to know the number of neutrophils that the patient has, and the absolute neutrophil count is one of the most common blood tests ordered in the clinic, and how much is discussed about neutrophils in books, like Clinical Immunology book, 7 out of 1,400 pages are about neutrophils. So I thought this imbalance between the emphasis in the clinic, the fact that we have 60% of our white blood cells are neutrophils, but then when you read about neutrophils, you only find 0.6% of the pages in the immunology book about neutrophils. I say, well, that's a big imbalance. I think that's an opportunity for learning more about neutrophils. Why is the body and the physicians are putting so much emphasis, whereas the immunologists don't? So these are the good actors for the movies that you'll see shortly. And then these are the bad actors, microbes that sometimes invade our body when we have a wound or some other conditions, and they proliferate and create infections. So one of the most common is a bacteria called Staphylococcus aureus. And you see here's bacteria shown in this purple, and the scale bar is 1 micron. So the bacteria is less than a micron in size. So if you think about the hair being 100 microns, this is really, really tiny. It's 1/100th of a hair. Not all microbes are this small. There are other microbes like Candida albicans, which is a fungus, which is a little bit larger. It has 2 shapes and it can morph from one shape to another. It has a yeast form about 3 microns, and it has a hyphae form, which is elongated, and that is can grow up to hundreds of microns. And then on the extreme side of microbe sizes, there is Aspergillus fumigatus, which is another fungus. It enters the body as a spore that is about 2 or 3 microns in diameter, but then it can grow, if it's not neutralized very quickly, it can grow into this longer hyphae that are hundreds and hundreds of microns long, or form these bodies that are filled with thousands of spores. And I think for engineers, what is most interesting is how we build these microscale arenas where you can look at how the white blood cells interact with these different microbes. And this is a picture from our research lab at Massachusetts General Hospital. It shows a clean room and people working and dressing up in a very clean environment. Because here we make devices that have very tiny features. And here, like, for example, we make channels that are 6 microns in width and 3 microns in height to look at neutrophils. And this is one of the changes that technology can bring to a field. This is how, on the left, you have how biologists are traditionally looking at neutrophils on flat surfaces. They have a micropipette with a chemoattractant at this end, and the gradient is guiding the neutrophils towards this chemoattractant. But you can see in this migration, the cells are moving, they stop and go, they change directions very often. So if you want to characterize this process of neutrophil migration, you have to use a lot of statistics, and your measurements will have a lot of noise. So the precision of the measurements and the comparisons you can make is reduced by this high level of noise. So then in the middle, look at what happens when you put the same neutrophils towards the same chemoattractant, but now instead of moving on a flat surface, they have to migrate through a channel. You see this neutrophil is moving at constant speed, very fast, without stopping towards the chemoattractant. So now the velocity can be estimated with very high precision, and comparisons between different neutrophils in different conditions are much more precise with less noise. And because we make the channels in our clean room, we can make channels that branch, and we can look at how neutrophils take the shorter route or the longer route towards the chemoattractant at the end. And what we find is that for neutrophils from healthy young individuals, 99% of the times the neutrophil will take the short route towards the chemoattractant, which means that they are very sensitive to small differences, and they figure out that the gradient is steeper on this side versus on the other side, and that's how they go. The difference between what is traditionally done on the left and what we can do on the right with the microfluidic tools, where here neutrophils can take any direction they want. Here they're restricted to either left or right. It's almost like the difference between digital and analog in electronics. Here with analog, you have a lot of noise. Here is digital, you reduce this noise and increase the precision. And we can take this idea of looking at neutrophils through mazes to an extreme. And this is one of the things that we did in 2016. We made this very fancy mazes, and we asked biologists around the world to send us cells and test which cell is the fastest to finish this maze. And you can see it takes less than 1 hour for neutrophils to go the distance, which is about almost 1 millimeter long, and end up in this chamber. And you can read more in this paper about what we did. But the main reason why we did this, and including this challenge, was that we wanted to find new tools where you can compare neutrophils between healthy individuals and neutrophils from patients that have a higher risk of infections. And this is one example of neutrophils from healthy individuals. They move very fast. You see in 2 minutes, they start moving. They go along the channels towards the chemoattractant along the shortest path. On the bottom, you have neutrophils from a patient that had a major burn injury. And you see that after 50 minutes, there is the first neutrophil coming in. And even that neutrophil doesn't go towards where the chemoattractant is, but it turns around. So if these neutrophils were the neutrophils to help you protect from infections, these neutrophils would be quite inefficient. Also on the technology side, just to wrap up with this part, we have now devices that can use whole blood directly. So we don't need to isolate the neutrophils. We can just take them directly from whole blood. And see here, the whole blood is compartmentalized. There are some small channels through which the neutrophils can enter, but neutrophils from a healthy individual wouldn't migrate unless there is a chemical signal attracting them, whereas neutrophils from patients with sepsis, for example, are less disciplined. They just leave the blood droplet and they go and wander through this maze of channels back and forth. And we can quantify this random spontaneous migration of the neutrophils and have a metric for the patient neutrophils and diagnostic in infections. And this is a paper that we published recently. And then now that we have these tools where we can look at neutrophils with high detail, when we put together the neutrophils and microbes, we can get the most detailed information that is available today of how this interaction happens. And what we build are arenas about 200 microns in diameter. If you think about the Roman gladiators, that's the model that we try to implement this at microscale. So we have these small arenas, 200 microns in diameter. We fill the arena with microbes, and then we put neutrophils on the outside, and the neutrophils can go in through this small gallery type channel, and they enter the arena and they encounter the microbes. And this is a paper published in Lab on a Chip very recently. And what we find is quite surprising. For example, we have Staphylococcus aureus, that tiny microbe that I showed you before, that is staining green into these arenas. And this is a live microbe that can grow and develop. And the neutrophils are on the outside, shown here in blue. And you see at the beginning of the movie, all the neutrophils are outside, the microbes are inside, the neutrophils are coming in. They come in both chambers. There's same neutrophils, same microbes. But here, because you have a little bit less neutrophils coming in, they proliferate so fast that they overcome the neutrophils. In exactly the same starting conditions on the right side, we have more active neutrophils. They clear all the stuff. This neutrophil ingested quite a lot of stuff. It cannot digest all of it. There are other neutrophils coming to the rescue and dividing this cluster of microbes. So there are a lot of interesting processes that happen that makes the outcome of this interaction between neutrophils and microbes less predictable. Especially with this microbe, Staphylococcus aureus, it has several defense mechanisms against neutrophils, and that's why this balance is broken very quickly. Along the same lines, we looked at interaction between neutrophils and Aspergillus fumigatus. And here we fill the chamber with Aspergillus shown in red, and then the neutrophils come in. This is an older movie, we didn't have all the colors, but you see that the neutrophils ingest the Aspergillus, and everything is phagocytosed. It's inside neutrophils. You think that the neutrophils have one, but actually what happens about 10, 15 hours later, the fungus that was ingested by neutrophils escapes and proliferates and grows into this long hyphae, like in a scary movie where the alien emerges out of a body of a host and starts invading around. We see significant differences between the neutrophils that we collect from healthy individuals, which are 99.9% efficient of containing Aspergillus, versus neutrophils from patients after transplant. And this is a movie with neutrophils from a patient. They're not able to contain this fungus as you would expect. And finally, one more movie before I stop to take questions. We can look in great detail of how neutrophils interact with the fungus. And there are some very interesting aspects of this interaction. You see a hyphae of Aspergillus growing through these channels from left to right, and then neutrophils coming from right to left and engaging this fungus. If there is a small number of neutrophils coming in, the fungus is branching, and then these branches can escape and travel through the channels further. If you have a lot of neutrophils, like you see here at the bottom, there is a higher chance that neutrophils will slow down and even stop the growth of the Aspergillus. The interaction between neutrophils and Aspergillus happens very fast. So in about 10 minutes, the Aspergillus can grow multiple heads and continue moving. And the neutrophils have to react quickly, and they have to react in large numbers. If you have no neutrophils, this is how fast the fungus is growing. If you have less than 10 neutrophils, you reduce the speed of fungus growing in half. If you have more than neutrophils, that's when you start having an upper hand and the speed is reduced about an order of magnitude. So I'm going to stop here for 2 minutes and I'm going to take questions. And I encourage you to ask any questions you have up to this time.
Hi, Daniel. I have a question. So in that first movie you show where you built the arenas?
Yes.
So the left one was failing to contain, whereas the right one was containing the microbe. Was there a difference between the neutrophils you applied in those?
No, that's a great question, and I couldn't emphasize enough. You have the same neutrophils, the same microbes, the same concentration of microbes in each chamber. It's a stochastic process because it's unstable equilibrium between the microbes and the neutrophils. It's either one or the other wins. You know, it's like the elections in the United States. Yeah. There's no middle ground. It's either one wins or the other wins. So in one case, the staph proliferate a little bit faster. Neutrophils were a little bit slower to enter. And that's why they microbes won and neutrophils lost.
Mm-hmm. Mm-hmm. There are a few questions.
Yes, so why choosing neutrophils, Jana? I think that's a great question. And if you go back to the first slide, to me, it was a big difference between how much physicians would want to know about neutrophils, their numbers, and how they are doing. The fact that we have 60% of white blood cells being neutrophils on one hand, so a lot of emphasis on the medical side and what the body does. And then the immunology books didn't pay much attention to neutrophils. And I think the reason is neutrophils are very robust cells, and in healthy individuals, it's quite boring to study neutrophils because they always behave perfectly. I think what is not known, that's where our tools help push the frontier of knowledge, is what happens in patients. You have individuals that are healthy yesterday, they didn't have any genetic mutation, but their neutrophils the next day after a trauma or an injury or an infection, their neutrophils are no more behaving normally. They have a distinct phenotype. So measuring that is what intrigued us. How do I introduce Aspergillus to neutrophils in microfluidic chambers? There are some very neat tricks that we figured out. And I encourage you to read the paper in Lab on the Chip, for example, and send me emails. What we do is we start with devices that are empty, that are filled with air. And when we prime the devices, we prime them with a suspension of microbes. So the microbes will enter the chambers that are the arenas. And then we wash all the other microbes that are outside the arenas. And we can do that thoroughly because whatever is trapped inside the arena is protected. Everything else that is outside is not. And then after we wash all the microbes, we introduce the neutrophils in the outside compartment. And then we take advantage of the fact that neutrophils can move, they can figure out where the microbes are, and they enter from the outside into the arena. Okay, so we'll move forward. We have one more questions that will answer the second break or at the end of the talk. So one of the things that was quite unique about neutrophils that was not known when I started studying neutrophils, and that was discovered around 2013, so 7 years ago, is this experiment that was reported in the Nature paper. Tim Lammerman and Ron Germain from the NIH looked at human neutrophils that they inject into a mouse. And for that mouse, they induced a small injury using a laser. So this is the injury that they make with the laser about 50 microns in diameter in the ear of a mouse. And then they watch the neutrophils coming together very quickly around this injury and sealing off the injury from the healthy tissue. And this process happens in very quickly. It's about 20 minutes to form this swarm of neutrophils. And what made this observation unique is that by the detail of in vivo microscopy, they could see that there was a scouting phase where neutrophils were moving around. When the first neutrophil would encounter the wound, it will apparently signal to other neutrophils around it to come together towards that wound. And that's how the swarm forms. And the group has figured out that there is a positive feedback loop happening between neutrophils that are the first to arrive at the site of injury and then also the infection, other studies shown, and the other neutrophils. So the neutrophils arriving at this site here, they produce a molecule named leukotriene-B4 that is a chemoattractant for other neutrophils in the surrounding to come to that site. But once those neutrophils start arriving, they produce even more leukotriene-B4. So every neutrophil coming to that site produces LTB4. So that increases the amount of LTB4, it stimulates more neutrophils, and more neutrophils are coming, and more leukotriene-B4 is being produced. So for the engineers in the audience, I think it would be very easy to understand that this is a prototype of a chain reaction or a positive feedback loop, where the input stimulates the output to increase the input. So neutrophils coming to the site of injury or infection produce leukotriene-B4, they stimulate more neutrophils to respond and migrate to that site. And those increasing number of neutrophils produce even more chemoattractant LTB4 that stimulate more neutrophils. So you have this positive feedback loop that is activated. And that's where the Nature paper that describes swarming stopped. And we scratched our head because he said, well, if this was true and it was the only explanation for the swarming, all the neutrophils in the body will be engaged into this action. So the 25 billion neutrophils that we have in the body, they will all come to the site of an infection, leaving the rest of the body unprotected. But that's not what happens. The swarming stops spontaneously after half an hour. And I think here, just to emphasize this chain reaction and why it's so important to have a good stopping mechanism, think about other chain reactions that are being used in practice. And I listed here a few examples, and I think the most relevant for an engineering audience is a nuclear reaction. You have an atom splitting and releasing neutrons that split more atoms. So you have the prototype of a chain reaction in the form of a nuclear explosion. However, 50 years ago, we figured out that we can very well control this chain reaction in a nuclear reactor, for example. So this relation between input and output can be controlled. And that's what we set up our minds to do, figure out how is this chain reaction between neutrophils controlled such that not all the resources are being depleted when we have a small infection. Okay, so how do we control the chain reaction? Now, the experiments that were done in the animal were in many ways heroic experiments because you would run one swarm per animal, and preparing the animal and also in vivo microscopy would take half a day. So to gather data, it will take a lot of work. And we said, well, maybe we can scale this up if we do the experiments in vitro. And this is what we did. We made arrays of microbe targets, microbe-like targets, zymosan particles, which are inert particles that will stimulate the neutrophils to form swarms. So we make this array of targets on a flat surface, and we have thousands of this. The way we made this is through a stamping technique. So we make a stamp out of an elastomeric material that has this tiny posts about between 5 and 100 microns. And we wet the stamp using a polymer solution that is negatively charged. When we stamp this pattern on a glass slide, we leave behind areas of polymer and areas of clean glass. So when we put zymosan particle on top, they will adhere on top of the polymer, but nowhere else. So that's how we make this pattern. When we add the neutrophils on top of this, we see swarming. in vitro now. So these are human neutrophils. Red are the zymosan particles. You see how the first neutrophils, the process starts slow, very small number of neutrophils, and then there is this explosive phase of positive chain reaction, and then everything stops at around half an hour. We can make a lot of these swarms, and this is scanning electron microscopy. At the end of the swarming process, we see swarms of equal size, equally spaced. So now we can increase our throughput orders of magnitude compared to the in vivo experiments. We can also combine microfluidics with simulation tools, and here is a tracking of real neutrophils moving towards a real zymosan target, and overlaid on this is the production of leukotriene before from the cells of the swarm. So you see how the production of leukotriene before starts small, and it starts engaging more and more neutrophils at a larger and larger distance. And this is the time in seconds. So this is about 10 minutes after it starts the chain reaction. We have hundreds of neutrophils already aggregated on the target zymosan. So if this was a microbe, it would have been completely sealed off and neutralized before it has a chance to proliferate. So getting back to the initial goal of this study, we said, well, if leukotriene before that was reported is a stimulus for this swarming, if we inhibit the receptors for leukotriene before that neutrophils have, using BLT1, BLT2 antagonists, we would expect the swarming to be stopped. But actually what we find is, so this is dynamic of a swarm in controlled conditions. When we start inhibiting the receptors, we see smaller swarms that grow a little bit slower. If we inhibit just the LTB4 receptors, the effect is significant, but the swarm still forms. So then we said, well, maybe there are other molecules involved in swarming, so inhibited additional receptors. We removed the serum and the complement factors, so we were getting smaller and smaller swarms. And what this showed us, and it confirms the findings from the in vivo experiments, that there are multiple signals that contribute to swarming in a redundant way. So if you take one out, there are other signals that still makes the swarming happen. So this is such an important process for controlling microbes that there are several redundancy systems built behind the chemoattractants. So when we saw this, that by inhibiting, leukotriene before, interleukin-8, and removing complement factors, we still have swarms. We said, well, we may need to take a broader approach to this problem. So what we did, we collected the supernatant from on top of the swarming slides, and we did a multiplex ELISA assay where we looked at 400 cytokines. And the reason why we were able to do this is we have, like I showed you before, we have thousands of swarms that happen on the same slide. They also are all synchronized. When we add the neutrophils to the slide, all swarms start forming at the same site. That is something that you couldn't do in vivo, and it proved very important here. And we compared neutrophils that are resting on a flat surface with no target, small targets, whereas neutrophils just phagocytose those, but they don't swarm, and neutrophils swarms around larger targets. And what we found was that for most of the cytokines that we tested, there were no significant differences between swarming and phagocytosis versus resting. There are a lot of cytokines being produced by the neutrophils when activated by either phagocytosis or swarming, But there were about a dozen that were quite specific to swarming, much larger amounts released during swarming than during phagocytosis. And we can look at this release in time, and you can see that the concentration of these different signal proteins, galactin-3, adipsin, nitrogen, they increase very quickly in the first hour, they reach a plateau. So that correlates with the dynamics of swarming. All these molecules that we detected are contributing to activating neutrophils and other cells. None of them is an inhibitory signal. So when we thought more, I said, well, leukotriene-4, the molecule stimulating neutrophils swarming, is a lipid mediator. So maybe we should look at more lipid mediators. Maybe the inhibitory signal is one of these. So here's an assay that we did in collaboration with Dr. Charles Sirhan at Brigham and Women's Hospital here in Boston. Using mass spectrometry, we looked at the supernatant and we looked for small lipids. And we found that LTB4, the signal for swarming, is produced with a peak at 1 hour. And then there is an offset in the production of an inhibitor, lipoxin A4, which is another lipid mediator derived from the same molecule as leukotriene-4, but which has inhibitory action on neutrophils. There are some other lipid mediators, resolvins and prostaglandins, that were identified in this study. So it looks like all the inhibitors are just a few, and they are all lipids, not peptides. We can verify this in vitro very easily by adding a large concentration of lipoxin at the beginning of swarming, and you see here swarming growing in control conditions, and when lipoxin is present, that swarming doesn't grow at all. So this confirms the role that lipoxin has. Why are all the cytokines being released? We think that they are helping not just the neutrophils, but interacting with other immune cells, like T cells and monocytes and dendritic cells. And they prepare the tissues also to protect them better against infections and inflammation. So with this, we think that the neutrophils are at the center of the immune response, and the swarming around clusters of microbes is a key event for triggering the more sophisticated mechanisms of immune protection involving B cells, antibodies, recognition by T cells. And that's confirmed by a recent study that showed that swarming is essential for the vaccination process to engage acquired immune responses. So I'm going to stop here to take a few more questions about swarming, and then I have about 10 slides more, talk about some medical applications of these tools that we developed and new findings. So please type your questions, and I'll take 2 or 3 minutes to answer them.
Daniel, there was one question from last time remaining. Maybe we can start.
Yeah, it looks like everyone is confused about swarming and chain reactions. I think when I mentioned the viral videos, I think everyone remembered that, oh, I have to watch that YouTube video and then share it with friends and initiate a chain reaction around that. Yeah, so I have a question. Velocity profiles from different cases, including burn and sepsis, very common mechanism that explains the behavior of neutrophils. Well, yes and no. So neutrophils, regardless if we are healthy or diseased, they use the same mechanism for migration. They polymerize actin at one end. They engage molecules from outside the cell through receptors, and there are different pathways for signaling. I think what happens during disease is a connection between the sensing elements, the receptors on the cell, and the mechanism motility are being disturbed, and neutrophils cannot follow signals properly, like you've seen in the movie with the burn injury. And some neutrophils are moving spontaneously when they actually don't have a signal that they should be moving. And that's another type of defect. So I think with these tools that we have, we can start dissecting those defects. Okay, so no questions about the swarming. Well, I think I'm going to go very fast towards the next phase because I think I lost my audience. So now what happens in interaction between neutrophils and microbes when the microbes are present not just in tiny individual microbes, but microbes that eventually grow into larger clusters? And to answer this question, we made swarms that start with live microbes. The same way as we put the inert thymosan particles on slides, we can put live Candida, shown here, in the form of yeast. And if we watch this over time, we see the Candida growing and forming large hyphae. So in 16 hours, you go from this phase of little spores to this phase of huge ball of hyphae. So what are neutrophils? What those neutrophils can do against this? So here's a movie showing Candida growing over time. Here's the time in hours. So at 4 hours, you see that the small clusters is now growing hyphae, and these are growing and growing in size. If we, on the other side, if we have neutrophils, you can see that the neutrophils would get very quickly, in less than half an hour, around the initial cluster. They'll cover the initial cluster, they'll form a tight seal around it and prevent the Candida from growing. It takes more than 12 hours before even one hyphae can emerge out of this compartment of neutrophils. We can quantify the area of Candida growth. This is green is unopposed Candida, and blue is when neutrophils are present. This process of containing this yeast, this fungus, is dependent on the neutrophil numbers. You have to have enough neutrophils to form good swarms. Otherwise, the size of the Candida cluster is less controlled. If we use fluorescent dyes, we can gain more insight into the swarming. And here's one part of the study that we did showing how when you start with different concentrations of microbes, so here you have about 20 yeast cells on the surface, and here you have more than 100, you get different responses from the neutrophils. When you have just a few isolated yeast cells, the neutrophils would crawl on top of them, they'll phagocytose, they will neutralize the target before it has any chance to grow, growing. When there are clusters of microbes, two things happen. So the microbes in clusters, they have this quorum sensing mechanism and they are becoming more aggressive, but the neutrophils have a response to that. They aggregate together and form clusters of swarms. And for an intermediate number, these swarms are going to be transient. They'll form, they'll destroy all the microbes, and then neutrophils are going to dissipate. If you have larger clusters of microbes, neutrophils are going to form a stable swarm. It appears about 2 hours that this swarm is contracting and compacting as a Candida, or maybe some of it is being destroyed and that's why the size is decreasing. And then later on, so neutrophils do other things towards between 4 and 6 hours. They do a very interesting thing, which is the neutrophils that are the closest to the microbes, they undergo a transformation where they release the chromatin from inside the nucleus and form a very toxic capsule around the microbe. The neutrophils outside the swarm are protecting the healthy tissue from the toxic chromatin-based substance that includes a lot of cationic proteins, a lot of enzymes like elastase, and a lot of oxygen species. So this is how we can look at this process in dynamic. In bright field, we can see the neutrophils forming a swarm around Candida. We can stain the nucleus of the neutrophils, and we can see the swarm. And then when the chromatin is being released, the fluorescence decreases because the chromatin that is stained in this slide becomes more diffuse. If we use a specific dye for free chromatin outside the cell, you see that it takes about 2 to 3, 4 hours before the chromatin is being released. But once it's released, it stays around the cluster of microbes. If we use enzyme to degrade this chromatin, the ability of the neutrophil swarms to contain Candida and prevent it from growing is decreasing. So higher concentration of DNAase, the more Candida can grow. And what this tells us is that this transition of neutrophils from being intact to the release of chromatin is a very important process. And this is one of the hot topics today in the neutrophil field. How are the neutrophils releasing chromatin? It's a process called mitosis. We can look also at the biochemistry of these swarms, and we can use inhibitors like for reactive oxygen, and we can see that swarms that otherwise would be nicely defined, they actually start falling apart when we inhibit the reactive oxygen species. You see here swarm forms and then they fall apart. So this gives us so with these tools that we have now, we can dissect not just at molecular level what the different molecules inside neutrophils do, but also at the biochemical level, what fractions are important for the control of Candida. And we can put this nicely on maps. This is a paper that we published this year in Nature Communications. You can read more about neutrophil swarming controlling Candida. There's an initial swarming, there's a release of nets, and then hours later, the Candida could escape. And that's a significant delay. Candida without the neutrophils to germinate and form this long hyphae in about 2 hours, whereas with neutrophils, that process is delayed by at least 10 hours. And this is in vitro with just a small number of neutrophils. So where is this important? If we look at neutrophils from patients that have an immune defect, like chronic granulomatous disease, where neutrophils are not able to produce reactive oxygen, we find that the swarms that these cells, these neutrophils can make are larger in size, but less effective in controlling Candida. And this could explain the higher rate of infection in these patients. When we look at neutrophils from patients after burn and trauma injury during sepsis, we find another interesting thing. We find that the size of the swarms, which I showed you in the previous slide, is very uniform when you have the same size targets, becomes very heterogeneous. This is a picture showing swarms with neutrophils from patients with sepsis. And you see that around some targets, there are very large swarms. Around other targets, there are no swarms at all. And this is shown in detail here. Swarm, on this target, there is no swarm. This heterogeneity, we think, may explain why the body is less effective in controlling infections in these conditions, like after trauma, like a car accident. Why is the rate of infection much higher in these patients? And by knowing this, there is hope that we could control and maybe restore the neutrophil function in patients where this function is perturbed. And one of the findings from the paper that I showed you with Candida was that there are factors like colony growth factor that acts on the stem cells in the bone marrow, but also on the neutrophil circulation to stimulate neutrophil activity. This is a time course of a patient at Massachusetts General Hospital, which every of these red bands signifies an infection where the patient had to be admitted to the hospital and treated with antibiotics. So you can see that every couple of weeks, there are several episodes of infections. And the physicians couldn't figure out what is the defect that this patient has. In collaboration with one physician scientist who's also an infectious disease specialist who was treating this patient, looked at the neutrophils. We found that these neutrophils form swarms that are less effective in controlling any microbe that we try, staph and candida. So this is a situation where we put, where we tested in vitro one of the drugs that could be administered to the patient, colony stimulating factor, which is used to treat usually a patient after chemotherapy. And we found that this can restore the ability of neutrophils to control microbes. So these arrows here now signifies the injections of this drug to the patient. And we can see that the frequency of infections was dramatically reduced. And this was just the beginning of the therapy. Now this patient has been observed for more than 6 months, and he didn't come back to the hospital with any other infections. So we think that this sort of tools, when put in the hands of physicians, would actually help more than just counting the neutrophils. And I'm going to stop here, and I think we have probably 5 more minutes to take questions. There are several people to acknowledge that contributed to this work, and many of them were postdoctoral fellows in our lab, several were physicians, some of the funding sources that funded our research. And if you have questions after this talk, I'll just put my email address here. You can ask questions. I also have a Twitter account if you want to make your questions public and maybe stimulate some discussion around neutrophils. Just follow this address and post your question in a public forum. So I'll stop here and try to answer your questions. I hope you enjoy the movies, and I'll be very happy to answer any question. I understand that many of you are not familiar with cells and microbes, so any question you have that will help clarify is very welcome.
Thank you, Daniel. It's always nice to hear you speak about your work. There are a few questions, if you can address them. I have a few of my own, but I'll wait until.
Okay. Yes. So I'll answer these 2 that are listed here, and then. So Darren asks, do you see 1D swarming in the gallery of arena chips or in the narrow channel devices? Yes. Does the swarming change with dimensionality? Happens faster in 3D versus 2D. So all the devices that we have are pseudo 2-dimensional. Because of imaging constraints, we want to have cells moving on flat surfaces, right? And the answer is yes. Swarming happens in other devices than the ones that we build purposefully for studying swarming. They happen in arena devices, they happen in long channels. It's one swarm here and there. If you want higher throughput, you build the best tool for doing that. Also in the arena devices, for example, we see swarms. They are not synchronized when they start because the conditions for triggering swarms are different in different arenas. So yes, swarming is a very robust thing that happens all the time. second question from JW. How do neutrophils determine the number of cells attack a pathogen? Do neutrophils exhibit quorum sensing activity on their own when encountering pathogens? This is a very clever question, and thank you very much for asking this. Yes, it appears that neutrophils can optimize their numbers against the targets. If you have a small target, you have a swarm that is composed of a small number of neutrophils. If you have a huge target that is hundreds of microns, like a big hyphae, you have hundreds of neutrophils that are recruited. It's a great question. We don't have an answer. How are the neutrophils knowing when to stop? It may have to do with a number of layers of neutrophils that cover a target. So it's like you have a larger object. The layers are the same, but the surface that they cover has to be adapted. Maybe when you reach the third, fourth layer of neutrophils, there is no diffusion of the signal from the inside. This target is sealed, and that's how this number of neutrophils is controlled. Zulal Mungali asks, I was wondering that what kind of environment is required for neutrophils to attack on microbes? Well, you'll be surprised. So in biology labs, when you run experiments, you learn that, oh, you have to run them on 37 degrees Celsius, you have to have 5% CO2. Neutrophils appear to be independent of all these restrictions. We have run experiments with neutrophils at different temperatures, and we find that neutrophils can work in the range from about 20 to 40 degrees Celsius. So that covers room temperature up to fever conditions. Neutrophils can function in a media that is void of any nutrients because they have their own storage of glycogen that can be converted into energy for the neutrophils to function. So if you run experiments in physiologic serum with no proteins, and you have microbes that don't have the resources to proliferate, neutrophils can still perform their action there. And yes, neutrophils can function in conditions of hypoxia, and apparently they even function better when there's low oxygen than when it has oxygen like a normal. So neutrophils are very well adapted to finding microbes anywhere, anytime. Amazing cells. Unal Akar asks, can the neutrophil swarming be affected by environment like a gel or other environments? Oh, definitely yes. We study swarming on a flat glass surface in a fluid environment, but in the body, you don't have, where is the steps, it is the exception when you have flat surfaces. Most of the time, neutrophils have to travel through extracellular matrix. They have to squeeze in between other cells. So conditions like the mazes that I showed earlier may be more relevant to what's happening in vivo. So are these conditions affecting swarming? Yes, you have probably more neutrophils that can travel towards the target faster if they come from all 3 dimensions than 2 dimension. It's definitely something that we would like to study and build some new tools to do that.
All right. Thank you so much, Daniel. I think the swarming questions came later. So,
yeah, I like this. These are very, very good questions. And I like to see that people understood and who are fascinated about these cells. As I told you before, I don't think they get the attention that they deserve for how robust they are. And, you know, as an engineer, you look and wonder how these cells can accomplish so much with, you know, them being just 10 microns in diameter.
Do you have any work to, you know, test out different drugs to improve the neutrophil function migration, swarming?
Yes, so neutrophils from healthy individuals, I think they are perfect. They don't need any help. I think the help that is needed is in neutrophils, for neutrophils that come from patients. Yes, some of the tools that we have could be scaled up to make, higher throughput testing beds. The restriction is always going to be the source of neutrophils from patients. You have to have everything ready at the moment's notice when the patient arrives, and you can test. You start using those neutrophils because those are the neutrophils that you want to correct. So it's not like the usual drug testing that you have some cell lines that you can use at any time. You plan your experiments in great detail. It's a little more challenging doing this for neutrophils. And yes, as I said in the last slide, with the patient with repeated infections, we have at least one compound that we know, colony stimulating factor, that restores neutrophil activities. And that's a great start because we know that it is possible, and there should be more drugs like that.
Mm-hmm. All right. So it looks like we have no further questions from our audience. I want to thank you once again for this wonderful talk. I think we've learned more on neutrophils and how they function. So thank you so much.
Thank you very much, Başak. Thank you, everyone.
Yeah. So to our audience, I just want to wish a happy new year because this was the last talk of this year. So we'll take a few weeks of break and resume the seminar series on January 6th in the new year with Professor Ali Koşar from Sabancı University. So hope to see you then, and Happy New Year again. Thank you, Daniel, again. Bye.
Happy New Year. Thank you so much. Bye-bye.
Bye.